Project One is really aimed at looking at biomarkers of exposure and biomarkers of effect. The biomarkers of exposure that we're looking at are the direct damage to DNA that chemicals can cause. Now, we have some very interesting chemicals that we're looking at because they're also essential chemicals in our body.
Our bodies are constantly being exposed to a wide variety of chemicals, from food we eat to stuff inside of our drinking water to lifestyle choices. Do you drink alcohol or smoke cigarettes? Also, we are exposed to toxic chemicals that are made naturally inside of our body. This is a natural process. Our DNA is constantly being damaged from a wide variety of sources, and ultimately these can lead to mutations, which can lead to cancer.
We need to know how much the exposure to environmental chemical is safe and how much environmental chemical is dangerous. The main question is which DNA damage inside or from outside, which has more impact for our body.
What we're trying to understand is what concentrations of certain chemicals increase the rate of mutations. Where does that difference lie? Where do the exogenous effects dominate over the endogenous background? Where does increasing concentrations ultimately have effects? My work is funded by a Superfund to help better understand the effects of these chemicals on people. Ultimately, help understand the regulations that might be useful in cleaning up Superfund sites and how dangerous these chemicals are to people who are living close to them or interact with the sites.
If we could find out the biomarker of DNA damage, then definitely we could monitor the population who are living very close to Superfund site.
And then we can use that kind of information to look at what is the ability of remediating a site that would have that particular chemical as a problem. What is the cost of remediating it? Would you rather be able to remediate 50 sites a year or 5 sites a year? It's going to make that kind of difference if we start having the science drive the issue.